Hello, everyone. My name is Hassane Mchaourab. I am a professor of molecular physiology and biophysics in the School of Basic Sciences in the School of Medicine at Vanderbilt University. Welcome to this edition of Lab to Table. We are going to pause for about 30 seconds to allow people to join. Okay, hello again, and welcome to those who have joined us in the last few minutes. Again, I'm the moderator of today's event, which will focus on a relatively hot topic about research ethics, broadly defined. And before I begin our discussions today, I will have a few introductory remarks, and I will start them with sort of an obvious observation from human history that science plays a central, pivotal role in the progress of society. Science permeates all aspects of our life today, the communications that bring us all together in a virtual world, in food production and food processing, and importantly for us here in the medical center, in the medical school, is in drugs and vaccines that we take to combat disease and pathogens. Since the 17th century, science advancement has been deeply rooted in what I consider one of the most important ideas of humanity, the scientific method. So briefly, what is the scientific method? It's a blueprint that guides the scientific endeavor in all fields, not in one field. It begins with statements about reality that are firmly rooted in empirical observations, whether these statements are about a black hole at the edge of the universe, a small molecule, RNA molecule, protein molecule in a cell. They are, as I said, rooted in empirical observation. They're not inspired by a set of beliefs. They're not written in some holy book. These statements then, sometimes we call them hypotheses, are challenged using rigorous and reproducible methods and approaches. The outcome of this challenge, of this testing that scientists engage in, could be that this hypothesis is our best statement about reality, about the world, about ourselves, about our cells. It also could be that the hypothesis gets modified, and sometimes the entire model underlying the hypothesis or set of hypotheses is revised. And human history is full of these glorious moments. They are glorious moments for all scientists, and I think the impact on society is remarkable. So today in our conversation, we're going to talk about these two aspects of science: the rigorous and reproducible testing of hypotheses, approaching them with a skeptic eyes. And we're going to talk also about the ever-changing nature of the models that scientists put forward. And both of these aspects have been thrown into the public discourse by the recent COVID-19 pandemics, where literally scientists were deriving models, updating models in real time, as they had to also weigh in on societal decision, on policy decisions that affected society in deep ways. So joining us today, a panel of experts that will weigh on this, and I yield the floor for them to introduce themselves. Matt.
Well, I thank you for the invitation, and I'm looking forward to our conversation today. My name is Matthew Schrag, and I'm a neurologist here, and I take care of patients with vascular diseases of the brain and Alzheimer's disease. And I run a basic science laboratory studying the role of diseases of very small blood vessels in the brain, capillaries and arterioles, and the way they contribute to neurodegenerative disorders like Alzheimer's disease.
Mark.
Good morning. I'm Mark Dennison. I've been at Vanderbilt since 1991, and I'm the director of the Division of Pediatric Infectious Diseases that treats children with complicated infections, but I also have a basic program in coronavirus research that I've had now since 1984, I guess, here at Vanderbilt. And we've worked on development of antivirals and vaccines.
Alan.
I'm Ellen Clayton. I've been at Vanderbilt since 1988. I'm in the Center for Biomedical Ethics and Society. I'm a general pediatrician and also a law professor here. And my work, uh, uh, the whole time I've been here really has been about the translation of research and the appropriate conduct of research and its translation to clinical care.
Well, thank you all. We're gonna just directly delve in, and we're gonna start with a question to Matt. And hardly any one of us does not know a loved one, a member of the family, an acquaintance, a friend, a neighbor who doesn't suffer, who hasn't suffered from a neurodegenerative disease, and definitely Alzheimer's is one of the most notable and common one. Matt, you will recently have weighed in a very public way on one of the paradigms that has shaped progress or has shaped research in this field, with consequence regarding federal funding and where the field moves, and definitely with consequence toward the development of therapeutic strategy. Could you please tell us about that and from the perspective of research ethics?
Yeah, I'd be happy to do that. It's sort of an uncomfortable topic, I think, for a scientist to talk about is research misconduct. We'd like to believe that this almost doesn't exist, that it's exquisitely rare. And I do think it's rare, but it happens, and it sometimes can happen in ways that has important consequences. So the stories I'm going to tell you today involve some work that I've done outside of my role here at Vanderbilt. And I think it's important to mention that when I'm telling you these stories, I'm speaking for myself and not on behalf of Vanderbilt University or Vanderbilt University Medical Center. In the late summer of 2021, last year, I was approached by a law firm and asked to consult for them on a research integrity concern. They had a number of clients who had raised concerns about a company developing a drug for Alzheimer's disease. And there were some questions about whether the data was accurate and whether there might have been some degree of manipulation in the basic science underlying the development of that drug. And I was asked to due diligence this concern and extend the forensic analysis of the data that underlie the development of this drug called sumifilin. And I undertook this project for a couple of reasons, and the most important one was that this drug was entering phase 3 clinical trials, meaning that about 1,750 patients with Alzheimer's disease are about to be exposed to this drug. And so if there are concerns about the reliability of the underlying data, as a physician and a scientist, there's a really critical ethical obligation to make sure that that's adequately addressed. And as full disclosure, I was paid for this consulting role. In the process of conducting this analysis, I found a pervasive pattern of surprising red flags in this data. And one of the techniques that was used extensively to understand how this drug worked was something called a Western blot. Now, not to panic, we're not going to get too technical, but a Western blot basically allows scientists to measure the amount of protein in a sample, in this case, brains, and learn some things about how that protein behaves. And the end result of that experiment is a photograph. And the photographs, to my eye, looked a little bit unnatural, in the same way that somebody looking at a magazine might think that a photograph had been retouched or photoshopped or airbrushed. I sort of had the same initial reaction, that there were elements of these experimental results that just struck me as looking unnatural. And there are some ways to forensically analyze these images to try to build confidence in that concern. And I assembled this report, and I extended the investigation past the boundaries of the development of this drug to look at papers that had been conducted by this same group of authors in other territories. And we found a very large collection of things that raised concerns about research misconduct. And I felt that this was substantial enough and involved a large enough amount of federal funding through the NIH and other venues that it needed to be addressed. And so I filed a report to the NIH about this. In the context of doing this, Very incidentally, I encountered another group of papers. I was looking at how other data scientists address similar concerns. And there was a paper from a scientist named Sylvain Lesnie, who works at the University of Minnesota, that came to my attention because it had a similar pattern of anomalies in the images. And because of the similarity, I had to look at the remainder of his papers. Now, Dr. Lesnie is not a hugely famous scientist, but he works very closely with some very prominent scientists. And one of the things that came to my attention was that a number of the papers in his body of work were massively important to the field of Alzheimer's disease. And one in particular was published in 2006 in the Journal of Nature, and is among the most highly cited papers on the topic of the basic science of Alzheimer's disease of all time. And depending on how you rank things, it's in the top 5 and might be the second most cited paper of basic science paper of all time on the topic of Alzheimer's disease. And the same pattern of image anomalies seemed to pop out of that paper to me as well. There were areas where there were sharp cuts in the background that looked like there might have been some cut and paste type cloning changes. There were areas of the images that looked duplicated in ways that didn't make sense. Some entire Western blots had been repeated and the samples had been relabeled to represent other things. There was a really large number of red flags. Now, it's very hard to get to a credible conclusion that this represents fraud purely from publicly available data. So we don't allege that. I don't allege that. But it certainly raises enough red flags to undermine my confidence in the data in that paper and in the body of work that came from that paper. And it raised, I think, questions for the field at large because of the importance of that paper. So that paper happened at a very important time for the field of Alzheimer's disease. The leading hypothesis had been that the plaques that accumulate in the brain and are made of this little tiny protein called beta amyloid. Beta amyloid plaques were the major driver of Alzheimer's disease. And that hypothesis had been around for 10 or 15 years and had really achieved dominance in the field. And for the first time, a number of clinical trials had been conducted that addressed that hypothesis directly and didn't perform terribly well. And there was beginning to be some questions raised about the reliability of this hypothesis. So this was a moment in the field where there was a major reformulation in the dominant amyloid hypothesis. And the thought changed from the notion that the plaques, these very large clumps of this beta amyloid protein, that these were the major drivers, and instead focused on much smaller clumps of beta amyloid, which were termed oligomers, and that these much smaller clumps of beta amyloid were responsible for memory impairment and neurodegeneration and not the larger plaques. And what Dr. Lesnie and Dr. Ash showed in this paper was that you can isolate one particular clump, a clump of 12 strands of beta amyloid, and it was particularly toxic to the brain. And you could remove it from the brain of mice that were mimicking Alzheimer's disease and transplant it into the brains of rats and cause them to have memory impairment. There's a striking connection between this hypothesis that oligomers were the toxic species and memory for really the first convincing time. The problem was, when we started looking into these concerns, it turned out that quite a few other people in the subsequent years had attempted to reproduce these results and weren't really able to achieve the same results. In fact, many couldn't even prove that these clusters of 12 strands actually even formed in the brain. So there were some questions about whether this aggregate called A beta star 56 even existed. And much of that concern never made it into the published literature, although a couple of scientists did publish papers saying they were unable to reproduce components of that conversation. So we came with this concern about the integrity of the underlying data and walked away with a much larger understanding of the amyloid hypothesis, particularly this oligomer reformulation of the hypothesis. But a lot of it had never been publicly discussed. And I think that slowed down the field and allowed this information to stay as a major part of the way we formulate and understand Alzheimer's disease for a period of 15, 16 years. And I think perhaps resulted in investments into a hypothesis that might have been better invested in some alternative targets.
Before I follow up, I just wanted to mention to the audience that please put your questions in the chat, and we'll get to them. There'll be set aside time to take questions from the audience. Matt, this was a great example of hypothesis and how hypotheses get tested and how hypotheses get reformulated. Where do you think the failure of the scientific method, of the failure for us to say, okay, done with this hypothesis, let's move on. Where do you think that originated from?
Well, I think that there's, as is often the case in situations like this, there's more than one problem, and that often it takes a conglomeration of issues. But one is that there is a priority in this field to discuss new, exciting, interesting findings. And when people try to publish data showing that they couldn't reproduce somebody else's work, that's just fundamentally not interesting, but it is fundamentally very important. And I think there needs to be a greater emphasis in reproducibility and making investments in understanding the reproducibility of core nodes of data, a real priority for a field, because I think it would allow that information to trickle out beyond the elite labs that talk to each other and sort of have a sense that this isn't working out. Allow it to trickle out to the troops, because a lot of important work is being done outside of these elite labs that have this insight into, the fact that certain things didn't reproduce. So I think that's very important, is a greater emphasis on reproducibility, and also the ability to publish reproducibility, the expectation that you publish reproduction of important nodes of data. I think it's also really important for us to diversify conceptually the work that's being funded. And I think that when there's bottlenecks in funding, it's very easy for a dominant hypothesis to gobble up all of the resources. And there's good reasons to invest in dominant hypotheses. Often they're dominant for a reason. But I think that intentionally maintaining some diversity in that pool of ideas is going to be very helpful. It creates a more competitive environment where people who are inclined to challenge dominant hypotheses are given a voice, so that these conversations are no longer viewed as heroic. This shouldn't be a heroic conversation. This should be an absolutely normal part of a scientific conversation.
Thank you, Matt. I think we'll revisit this aspect of science and science ethics, research ethics. But I would like right now to continue with the conversation and move it to something that has been in the news for 3 years and has had its share of controversy and definitely its share of myth and stories. This is, of course, around the COVID-19 and the ethical question that the trajectory of this infectious disease has posed. And I want to shift gears and go to Dr. Mark Dennison and talk about his perception of sort of development of vaccine and antiviral and the societal interest in understanding viruses and how viruses can escape detection by the human body, or can escape vaccines, and how the coronavirus story weighs in on this aspect and the ethics of it.
Well, thank you. First, disclaimers. I'm a member of the National Science Advisory Board for Biosecurity, or NSAB. So if people want to Google NSABB and look up recent conversations and things that have been nationally presented, they can see other comments by me and others about some of these topics. And also I'm multiply NIH funded. So these are my comments associated with certainly some of our research and some of my input I've had. We could talk about what Dr. Schrag has done for the entire time and for many, many hours, but I think just a few more topics and perhaps trying to approach it from the idea of how do we prospectively think about these topics? How do we prevent issues of these kinds of challenges from arising, and how do we deal with the issues of how do we validate? What kinds of experiments should we perform, and what kinds of oversight should there be at the level of the laboratory, the institutions, the NIH, and the public, and transparency to the public? And all of these we're dealing with simultaneously. I would have. Perhaps a simple, easiest question would be, if we're talking about risk or benefit, is the question I've had for about 30 years is, what good does it do working with a mouse coronavirus in a virus family that only causes human colds? What's the benefit of that? And why should US tax dollars be spent on such a project for so many years? And I think now the answer is, well, I'll tell you, in 30 years, when we've had a chance to use those models to develop a fundamental understanding of coronavirus biology that led to our initial testing of the drugs that became remdesivir and molnupiravir. And that was really based on very, very fundamental supported research that had to be highly reproducible over a very long period of time, and could lead to really longitudinal studies of the virus biology that then led to some of these compounds. But I think just really, I want to leave time for discussion. So a couple things. So the things that are arising now are, okay, we have a virus that's an incredible evolutionary machine, exceeded in exponential fashion anything I would have even anticipated in terms of the adaptability and change of a brand new virus coming into human populations. So I think as we talk about these ethical issues related to research, what we understand is the research is ongoing by the virus every day, 365 days a year, 24 hours a day. It's replicating in a human being or in an animal or in both. And it's the viruses that are here tomorrow are the ones that are successful today. So this evolutionary war is continuing. The research is ongoing. And what we're trying to do is get insights into it. We're trying to understand how it might evolve. Can we predict that? Are those changes that we see coming up, are they predicting something that's going to truly escape immunity? Is it going to escape the drugs and the vaccines that we've been working so diligently to try to develop? So these are all the kinds of questions we want to do, but let me get a little microcosm on just a couple topics. One is the issue of, now we have a drug like remdesivir. I won't go into the amazingly beautiful details about how it works, although I think you would find them fascinating. But I want to say that we know it targets a specific enzyme in the virus called the telomerase. And we have published studies showing how the virus might evade that. Those studies were done before the drug was approved. We were studying the fundamental mechanism. So the question is now, is if the drug is being approved and is being used, can we study how the virus might escape? Because we would do those studies genetically by passaging the virus in culture and looking to see over time, in the presence of the drug, how it developed mutations that might allow it to escape. Our goal of understanding that is to look at the fundamental mechanism of the virus, to target it better with other drugs, and to potentially make sure we're prepared if those mutations would arise in a population while we're looking at viruses that arise over time, naturally, or in the presence of the drug. That's our goal. But the questions would arise, is it okay to do that in a laboratory, to actually work on a virus, to test which particular mutations might arise? Is there a risk associated with that? And what's the benefit to society over time? And these are questions that are being struggled with at every level. How do we understand the virus further if we don't do those studies? Can we try to understand how a virus might escape from vaccines? Or can we even do surveillance and try to isolate and culture viruses from nature that might arise and cause pandemic in the future? So these are all questions that are really, really foremost in our minds. And as we go through them, they have implications not just for us, but for our trainees, because there's different kinds of risk and benefits. So some of the benefits are obviously our ability to predict, our ability to prepare, our ability to prevent. What are some of the risks? Well, the risks are, would we generate something that wouldn't be generated in nature? These are ones that people state. I have various feelings about these risks, but they're ones that people state. There's risks to those around who might get ill from them. There's other kinds of risks that aren't really considered societal risks, but are real. And those are that if we put up substantial enough barriers and obstacles to doing fundamental research, that we might never have investigators do it in the first place. and that we might have a whole populations of the workforce in the future that aren't focusing on these. And then when the next pandemic comes around, we'll be saying, why weren't we prepared and why is nobody studying this? So I think these are just a few sort of little launches across the bow, thinking about what some of our research ethical questions are. But I wanted to sort of raise those, and I'd like to move on and perhaps either address questions or come back to some of these topics.
And so what you touched upon the question of studying how viruses adapt, and can you speak to the type of regulations that are in place to protect? I mean, you've talked about but I think the public at large should know and should understand and appreciate the level of surveillance that is done in order to prevent sort of the mishaps.
Well, our oversight is everything, and we know your introduction on research ethics. I think the very first one always starts with the training of scientists and their individual commitment. We don't want to have what happens with Dr. Schrag have to happen, right, where someone else has to discover that someone has bypassed this. And this is about the ethics of the individuals and their commitment to reproducibility. I think there's a couple of things. One is a level that we need to do a better job of communicating overall why we do things. We need to be open to the idea that there may be some things we can't do just because we think they're important and interesting experiments, that there may be limits to that. Also, we need to be reporting more our negative data, right? So what did not work? And there needs to be motivations, and there needs to be incentives for people to openly and transparently report experiments that did not work and why they didn't work, and to describe it, because those in some ways will form a better fundamental basis. And if they're never reported, then people may repeat the same experiment over and over again because that negative data wasn't reported. So we need to make sure those incentives are in place. Second is those issues related to biosafety and biosecurity at the institutional level. So there are institutional biosafety committees. There are oversight committees that are to look at whether the path works with pathogens might introduce what's called gain of function. The word has bad connotations, but the positive performance is we don't want to do something that's going to give the virus the capacity to escape or to cause a pandemic if it might not have originally. Again, I think these risks are exceedingly small, but those oversights are in place. Third is the level of the NIH, which is at the level of grant review, where they would be looking, is this scientifically feasible? Does it make sense? Does it have value and benefit? And is it going to be done in a way that's ethical? And then at that level, then those would be flagged for the potential that there may be any concerns that this might introduce or change a pathogen in a way to increase its transmissibility or its disease, its virulence. Those then could be referred for additional review at the national level and by specific panels that would look to see whether there was a risk that seemed to exceed the potential future benefit. And we can talk about how that's balanced and who should be involved in that, and what kinds of public transparency should occur, what kinds of protections we put in place for investigators as well as for the public. So, so I think and then finally, there's agents that are called select agents. SARS-CoV is one of them. There's many others that basically have oversight and reviewed by the CDC as well, and specific requirements for biosecurity and biosafety, and that it require extraordinary oversight of investigators and individuals working with these. So I can tell you that we spend an extraordinary amount of our time being sure that at all of those levels that our work has been reviewed and has been deemed to be scientifically valid, to have potential benefit to society, and to have risks that can be either managed or mitigated, or that are so minimal as to allow their research to go forward.
Thank you, Dr. Dennison. I'm going to switch gear a little bit now to Dr. Clayton and sort of try to take these two examples and sort of abstract at some level the ethics implication. I'm going to give you free rein here to tell us about a lot of these ethical issues, whether it's in ensuring that the scientific community at large knows about the failure of particular hypotheses, and also the moral responsibility in ensuring that cost-benefit analysis happen in the cases of research on viruses and pathogens. You're muted, Ellen.
Thank you, and I'm delighted to join this conversation, of which these presentations have been incredible. Let me talk a little bit first about Matt's presentation. A fundamental failure in a case like this, it's hard to know without getting into talking with the actual people involved, but you can hypothesize what some risk areas are. One is that you have to believe in what you're doing, and that you may get too tied to a result that you think you've had that just gets in your way of having an open mind. Another thing to say is conflict of interest and conflict of commitment. And I say that in part because coming up with a big hypothesis often means that you get all kinds of things, including promotion, better access to funding, all of those sorts of things. I'm not saying that we can live without a conflict of interest or conflict of commitment. I think we have them. I think every person has those all the time, but you really have to be aware of them and you have to take them into account. And I think a fundamental thing that Mark said in our conversation yesterday is that when you are doing science, or indeed when you are advocating any position, you have to have an open mind to the possibility that maybe what you think you actually believe, or what the data actually show. I'm not talking about belief is not a science issue so much, but that you could be wrong. And that's essential. And if you don't have that open mind, you can get in real trouble. The second thing I would say that Matt pointed out is it is important to publish data that challenges the dominant hypothesis, because it could be that the dominant hypothesis is wrong. We used to think that having stomach ulcers was because of stress. Turns out it's an infection. I mean, who knew? And so, so those things always have to happen. The other thing to say is that actually calling out the dominant hypothesis can take courage. And so, you want to be careful, to make sure that you see a problem in the data. But it's important to be able to realize that suggesting that there might be something wrong is actually the scientific method. It's not an ad hominem, and it's not a wrong thing to do. And so, so I think that all of those things are really important for Matt's story. I think the information that Mark talks about is, I guess what I'm saying in science, we are always walking on the razor's edge. We have ideas about what we think the data show. There is a real, not zero, possibility that we are incorrect. That we do things that sometimes pose risks, and we do and have the superstructure to sort of try to assess risk and benefit, and to come up ethically and politically, and I mean small p politically, defensible guidelines about what research should go forward and what shouldn't. But in every aspect of that, we have to entertain the possibility that our vision and the vision of the overseeing agencies may be obscured, and they may be wrong, and it may turn out in the future that something that we thought was true was not true. But the important thing about research ethics is to be conscious at every level of the possibility that we may be wrong, and that factors may be getting in the way of our ability to see the truth clearly. That's what it's all about.
If I could just make a quick comment about that, Ellen, I agree 100%. This is something you deal with on the, the relationship between clinical medicine and research. Same principles apply. So, for example, if someone is presenting data to me about a patient with an infectious disease, and they'll present a series of ideas, and it's clearly directing toward a specific hypothesis. And then we're going there. And as we get there, my first question that we ask as a team is, what have we missed? How could we be wrong? How could we be missing the true hypothesis? And I think the same thing in the lab. And we're talking small science with a small, I'm talking science with a small s here, right? Which means that in terms of working with students and trainees and investigators in general, the first thing is I tell them, we're wrong about our interpretation. I just don't know how. And then I say, but the only thing we can do as investigators is we can be sure that our data as it's presented and reproduced is unassailable. That 20 years from now, when you look back and the interpretation of your work has changed, that data that you submitted, the primary data you generated, the approach you did would be entirely reproducible and the same result will be obtained. New data will change how that is interpreted. And I think this is an important point also for the public, is to understand that we should be engaged in this process. And COVID is a perfect example that it evolved so quickly and changed so rapidly that there was a perception in the public oftentimes that we were wrong. We kept saying the wrong thing. We changed our minds. The science wasn't really valid because the interpretation changed. This was just sped up, accelerated, both scientific evolution and viral evolution, where we were looking at things and they were changing. And so our interpretation had to change. The vaccines were fabulous, but they don't protect against infection completely, but they do protect against severe disease and death. Now we're getting variants that might have more immune escape. We're going to need boosters. Yes, we didn't think we might need them. Now we do. That is not flip-flopping. That is evolution of the science. And I think that's important. But the data that is used to base that on needs to be unassailable. And that's our ethical responsibility as scientists and mentors.
I want to follow up on this point, Mark, and particularly bring up the question for Matt, Mark, and Alan about communication. Now we have an interest, the public have an interest, society has an interest that science continues to be robust, continues to be funded. How can we communicate better to the public so that what we went through during COVID is not repeated? How can we, what level of communication scientists should be trained at, or I would like your opinions on this, all three of you actually. Mark, go ahead.
I'll let someone else start because I just
well, I'll say something about this. First of all, I think we need, one of the things that science has done to itself, and I have to say genetics has really done to itself, is, oversold what it can deliver systematically. And one problem in our current communication environment is that if it doesn't sell big, then it doesn't get talked about. On the other hand, I think we have to be really careful about saying that that what we know is an evolutionary process. The reason we do science is so that we know more tomorrow than we do today, and that we can make better decisions on the basis of that. But I think that what we really need to be saying in cases like COVID, for example, was that these are what our preliminary results are. It was a miracle to get those vaccines in that period of time. And it was a miracle in part because Mark had been working on coronavirus for decades. But just to say that this is our best shot on the basis of the best available evidence. We are trying hard to get even more evidence that will inform our decisions better. That's what you pay us to do. But we cannot guarantee that what we say today is going to be the same thing that we say tomorrow or in a decade. In fact, in a decade, we can be pretty sure it won't be the same thing we'll say. So I think on the one hand, we need to emphasize the role that data have to play in informing our judgments, at the same time saying that we don't have a vision of the truth at this point. And our path will vary depending on what the new data show. I think we really have to do that. And I think in some ways science has kind of hurt itself a little bit by sort of overselling the value of what we know now. I mean, what we know now is really important, but we're going to know more in the future, and we're going to have better paths in the future.
Matt, what you've talked about, of course, it has generated, I mean, the Alzheimer's story and the search for cure has been really frustrating. And there are hopes and every now and then about these. So could you speak about sort of the relationship between the veracity of the basic science and sort of these clinical trials and sort of the relationship, or how do we communicate this to the public also?
Yeah, well, I think this is a hugely important point because we have a special partnership with the public and with patients in this process, right? You're not going to cure Alzheimer's disease without a huge number of patients submitting themselves to clinical trials, and that comes with risks and uncertainties and a lot of hopes. And I think that that's a very special ethical obligation that we have to meet as scientists and physicians. Is that one, we're coming to patients and asking them to take the risk of a clinical trial when we have a reasonable expectation that the science supports doing that. And obviously it's imperfect and most trials will fail. And so, but we need to make sure that we give patients the best shot possible. And I also think we have an obligation to communicate with patients what we know and what we don't know. And to be really honest and candid about that. There are going to be failures in the scientific process. This is a profoundly human endeavor, and we're going to be wrong a lot. And I think that the robustness of scientific development comes because we acknowledge that and we address that. And as others have alluded to, that the science evolves and tries to self-correct. And that's why entrenched ideas are so potentially damaging, is because it can interfere with that ability of science to become, to be self-correcting. I also think that there is an important role for the public in oversight of this process. A lot of the ways we address research misconduct, which again, fortunately is rare, but is done very privately. And I think that's probably the right way to handle most of these things, is to very carefully investigate things and to do it privately until we understand what's happened. But there are times where the process creates some danger for the trajectory of the field or for a large number of patients, where we may need to break that protocol, that boundary of privacy around these potentially embarrassing issues, and speak publicly about them. I think it's important that any patient enrolling in clinical trials for this company, for instance, is aware of the concerns. Whether they agree or not is up to them, but we owe them a fair chance at understanding what their risks are when they sign up to participate in those clinical trials. So there are definitely times to do that. I found it was helpful to partner with a journalist who is very gifted at communicating these things to the public. And I think that those relationships can be very helpful. To have people who are professionals at communicating complicated scientific ideas without losing some of the nuance. Certainly there's some risk of that when when we communicate complicated things can get boiled down into very simple headlines, and that can be dangerous and damaging. But I do think it's important that we communicate our failures and our successes very candidly with the public.
So before I turn it to the chat, the public, yes, I'm going to turn it to Mark for a final thoughts on this topic.
Oh gosh, I don't want the final thoughts, but a couple of things. One is I think there's some, our need to communicate with the public. I think all three of us here have felt both the positive and the slings and arrows of trying to communicate in an educational way, that there are not always, there are other ethics to consider, not just scientific ones, but journalistic ones, in terms of what is communicated and what is extracted from the comments we might make, and that we need avenues that allow for a more thoughtful, long-term, direct communication. I think of something like TWIB, This Week in Virology, with Vince Racagnolo, who was has long conversations with scientists, and it allows to go into the details of what worked, what didn't work, what are the nuances of it that are in a forum that wouldn't be as widely distributed as something that's high impact. The second is when you look at grant funding mechanisms, and when you look at some of the guidelines for an RO1, which is a fundamental investigator-initiated grant, there's issues in terms of review. Is this paradigm changing? Well, is it going to change our model? Is it have the potential to have high impact? Well, I don't know. Always, a lot of what I do is very incremental, and I want to see how a specific protein works, and it will develop as I go. And if it creates an interesting phenotype and opportunity, I'll pursue it. But is it going to change the model and paradigm? I don't know. And everything can't be paradigm busting, because lots of things are actually right and need to be maintained and don't need to be changed. And then finally, I think we need to get away from words that are used in public and in a private, such as words like proof. This provides proof. And if a student stands up in a meeting and says, I'm doing this experiment in order to prove X, Y, Z, I say, let's take a time out for a minute and sit back. We're going to test that hypothesis, but the goal is not to prove. If your goal is to prove, then you're heading down the wrong track and you're going to get trapped. So I think that there's some things like that we need to do in terms of, and we don't control all the communication that goes on. So the best we can do is be very careful, very reproducible, and very clear in our communication, and not overstate the results of what we do.
Thank you. Thank you. I'm just going to get a few questions that have been put in the chat room, and the first one is directed to Matt, and it reads: you mentioned some very questionable data with substantial patient implications. In investigating this data, who ultimately makes the decision, and on what basis it's done, whether the data is whether the data is reliable? Is it the journal only? I believe in this case, the journal cleared the company. And if so, how much of a conflict of interest would that present, since they approved the initial publication?
Yeah, I think these are the right questions, and they are complicated questions. I think it would surprise people. It certainly surprised me going through this process how these types of concerns are often handled, and how often major conflicts of interest are present. For NIH-funded research within the last 6 years, the NIH does have mechanisms to review the reliability of data when there are concerns, and typically the NIH will refer this issue to the Office of Research Integrity at Health and Human Services, who in turn will refer it to the institution that the individual investigator works at. And that tends to be where the definitive investigation is done, is at the home institution by research integrity oversight boards locally. And there's some reasons for that. And it does appear to be a conflict of interest, because of course the institution receives a portion of the funds from investigators who are funded, and it's certainly some reason to clear investigators. But also, I think in fairness, the local institution is also the people who have the most access to understand the totality of the underlying evidence. And so absolutely this relies upon people having an honest mindset when these issues are addressed. There is very little regulation of publishers. And so publishers are not, and most of them are not answerable to oversight boards. And so the way they respond to these types of concerns is remarkably variable, and remarkably, in most cases, slow. I think it's not unusual for concern about research integrity brought to a publisher or a journal to take many years to address. And some are never addressed at all. And so I think that that's potentially a problem. And of course, journals do have, in many cases, conflicts of interest, because they did publish the original work. I think a lot of the progress that I would like to see would be to look at the role of the publisher more carefully. I think that when a journal or when a publisher stands behind data, they should have an obligation in perpetuity to defend the integrity of that data. And I think that when there are concerns, there should be an expectation, one, that they are addressed in a reasonable period of time, and two, that there is a reasonable transparency that there are concerns. Many of these papers can be under investigation for a protracted period of time without alerting scientists that there are concerns. And I think there's certainly a time for a journal to use a tool called an expression of concern, which is less than a retraction, but to alert people that there are questions and that there is an investigation present when those concerns are credible. I think that this tool can be used more broadly to make sure that this dialogue is as transparent as possible. But I think there's no doubt that this is an area that needs some work.
Thank you, Matt. There's another question, and it's open for all three of you, and comes and asks about how do we train scientists to accept being wrong, I guess to be skeptic on a consistent basis when researching a hypothesis. How do you build that resilience? Who wants to field this one? Mark?
Just a few brief comments. Like I said, we need to incentivize it, that it's a positive. It's oftentimes someone will come in and say, my experiment didn't work. And my lab sit down and plop down and give me the data. I'll say, let's look at the data. I say, oh, your experiment worked great. It just isn't what you expected. It's fantastically well produced and reproducible. Congratulations. But I think from their perspective, it's like, I can't move forward with my dissertation, or I can't move forward with my publication. What am I going to do now? So we need to have pathways that encourage that and begin to look differently about what constitutes scientific progress and productivity. I think that's one thing we need to encourage. We need to sort of almost celebrate those, because science is about 90% of that, or 95% of that, if you look across a career. And sometimes it's spectacular in terms of how much it fails and how rare it is to have something that is longitudinally reproducible and moves the progress forward. And that's where you also, the idea that if you get something that's very positive, very exciting, that's when you have to be at your most cautious and you're most careful in terms of your view of it. I think we need to incentivize ways to have cross-institutional reproducibility. So, for example, if I have a virus mutation that I think is incredibly important, that really quickly being able to find somebody in a consortium or a group or a mechanism, or just openly being able to, I'm really encouraging these, like what's happened with COVID, which is the open science consortia that are doing things in an open source way with data freely available online, multiple investigators. So how do we at institutions and at the NIH level and at funding levels incentivize that so that people get credit and so that they are able to share it? On the flip side, we need to be careful about predatory preprint servers that aren't really vetted. Now, bioRxiv and medRxiv, I think, are very reputable and they're very careful, but they also are publishing things that haven't been peer reviewed yet. And so I think we need to, that is here to stay. That is here to stay. And now there's places that do not have the same level of oversight as those. So people can get stuff out online anywhere, and it can have sometimes, in some people's eyes, the value of a highly reviewed publication. And that doesn't address the issue that Matt had. So that's, I think, those are some other areas that we need to be really aware of and careful of.
I'd like to add to that one of the things we should emphasize with our trainees is what we're trying to accomplish. We're not really trying to publish papers, and we're not really trying to get grants, right? Those are tools in the process, but we're trying to cure disease. And if you want to cure a disease, you have to be right? There is no shortcut. And so you can't twist a piece of data and reinterpret a piece of data incorrectly and expect to make progress towards understanding a mystery or understanding a disease mechanism. And so I tell my trainees, if you want to get rich, if you want a disease where you're going to be, or a job where you're going to be successful an awful lot, if you want power, there are much easier ways for people who are this intelligent to do that. You do not belong in academics if that's your motivation. The people who are here should be motivated to understand the mysteries of these diseases and to solve these diseases. And there's no way to cheat to do that. You have to keep your eye on the ball. And that's not a paper, not a grant. It's about making progress on understanding the disease process.
I want to follow up with this, and I think what both Mark and Matt are implying is that there need to be also a change in the mindset of how funding is decided. I think it may require that drastic of an action, both in terms of allowing negative results to be published and taken into consideration, as well as a faster turnaround and of grant reviews, as well as of implementation of some of the policies regarding dissemination of data and open science and open sources of science. So I think that's a challenge for the funding agencies. I think we're coming to almost the end of our discussions. Any last points that, Ellen? I see you unmuted.
No, I think that this has been wonderful.
Well, I'm the moderator. I get to wrap up. I think we made important points today, and I think one that stuck with me, probably because of my bias, is Mark's point about spending maybe the better part of his life researching viruses that he thought at some point, who cares, they just cause the common cold, only for that basic knowledge. But that basic knowledge at the level of molecules, at the level of proteins, at the level of fundamental evolution of viruses, to play a central role in saving lives, in having an impact on society. I think I also want to re-emphasize Matt's point that yes, science has the mechanism to self-correct, but sometimes the self-correction process can take way too long, and then in the meantime resources are being spent in what I would say off-highway dead-end pathways, to borrow an analogy from Mark yesterday. So that it's important for both the funding agency and the public to be engaged, but also for the public to appreciate how science, how the scientific method or the scientific process evolves in time. And I think with those thoughts, unless you have something to add to them, I would like to thank the audience and thank you for a spirited discussion.